Mutations are fascinating phenomena that occur within the genetic code, the very blueprint of life itself. At their core, mutations are changes in the DNA sequence that can arise from various factors, including errors during DNA replication, exposure to certain chemicals or radiation, and even viral infections. These alterations can have significant implications for an organism, ranging from benign to detrimental, and in some cases, they can even be beneficial, driving the process of evolution. To understand mutations, it's essential to grasp the structure of DNA. DNA is composed of sequences of nucleotides, which are the building blocks made up of 4 bases: adenine, thymine, cytosine, and guanine. The arrangement of these bases encodes the information necessary for building proteins, which perform countless functions in living organisms. When a change occurs in this sequence, it can lead to a mutation, and these mutations can be categorized into several types: insertions, deletions, and substitutions. Let's start with insertions. An insertion mutation occurs when one or more nucleotide bases are added to the DNA sequence. This can happen during DNA replication when the machinery responsible for copying the DNA stumbles and adds an extra base. For example, consider a simple sequence, ATTG. If an adenine is inserted, the sequence becomes AATG. This seemingly minor change can have significant consequences. If the insertion shifts the reading frame of the genetic code, it can lead to a completely different protein being produced, potentially resulting in a malfunctioning or non-functional protein. Such frame shift mutations can lead to serious genetic disorders, such as cystic fibrosis. Next, we have deletions, which are the opposite of insertions. A deletion mutation occurs when one or more nucleotide bases are removed from the DNA sequence. This can also disrupt the reading frame, leading to frame shift mutations. For instance, if we take the original sequence ATTG and delete the first A, we are left with TTG. This change can alter the entire protein that is synthesized from that sequence. Deletions can occur naturally or can be induced by environmental factors, such as radiation. An example of a condition caused by deletion is Duchenne muscular dystrophy, where specific deletions in the dystrophin gene lead to severe muscle weakness. The third type of mutation is substitution, where one nucleotide is replaced by another. This can occur through a process called point mutation. For example, if the sequence ATTG is changed to ATCG, the T has been substituted for a C. Substitutions can be classified into 2 categories: silent, missense, and nonsense mutations. A silent mutation does not change the amino acid sequence due to the redundancy of the genetic code. A missense mutation results in a different amino acid being incorporated into the protein, which can affect its function. For instance, in sickle cell disease, a single nucleotide substitution in the hemoglobin gene leads to the production of abnormal hemoglobin, causing red blood cells to assume a sickle shape and leading to various health complications. A nonsense mutation, on the other hand, results in a premature stop codon, truncating the protein and often rendering it non-functional. Mutations can be spontaneous, arising without any external influence, or induced by environmental factors. While many mutations are neutral or harmful, some can confer advantages, allowing organisms to adapt to their environments. This is the driving force behind evolution. Over generations, beneficial mutations can accumulate, leading to the diversity of life we see today. In summary, mutations are essential to the dynamic nature of genetic material. They can manifest as insertions, deletions, or substitutions, each with the potential to alter the course of an organism's development and function. Understanding mutations not only sheds light on the mechanisms of heredity and variation, but also opens doors to advancements in medicine, agriculture, and biotechnology. As we continue to explore the intricacies of genetic mutations, we unravel the very essence of life itself.